So in the second half of this chapter, we're going to be looking at the different types of eukaryotic microorganisms. And in this first section, section 5.4, we're going to talk about the fungi. So we're going to look at some of the general anatomy of the fungi, different terms like heterotroph, saprobe, and parasite, and explain the relationship between fungal hyphae and the production of mycelium, and different ways that spores arise in fungi, and then two detrimental and two beneficial activities. So from the viewpoint of humans. So the kingdom fungi has approximately 3 to 4 million species, and they can be divided into two basic groups, the macroscopic fungi and the microscopic fungi. And previous versions of this textbook, we went into much more detail on the divisions of fungi, but basically this is the way we're going to look at it here, because we really are looking at a systemic approach and really just need to understand what kind of organisms can cause disease and damage to crops and things like that. And so there's the mushrooms, puffballs, fungi that have gills, and then those are macroscopic ones, and you may use mushrooms in your food and your salads and pizzas and other Chinese food and stuff like that. So you may eat mushrooms. And then there are the molds, which cause decomposition to the fungi, and yeasts, which cause disease. So we're really, the microscopic versions are probably the ones that we're more concerned about as being harmful to us, but both can be beneficial. So the microscopic morphology means shapes. We have the yeast cells, which are round to oval in shape. They're not usually they're bigger than bacteria, but they may look more oval and not as regular in their shape. So they use asexual reproduction. That's the yeast cell, the single cell version of fungi. Hyphae are long filaments, as you can see here. This is what they look like on macroscopically when just looking at furry stuff. That's usually, you've got mold. So mold is the other type. So you have the yeast form and the mold form. And the yeast form is one-celled, and the hyphae and mold form is multicellular, or filamentous is really the way we would look at this and call it filamentous, because they don't technically form tissues. This is why they don't consider, and they're very different than plants and animals. So I don't really like the term multicellular for this. It's more filamentous fungi, which we call the molds. And so the hyphae are those single strands. And pseudohyphae are chains of yeasts. So they look like hyphae, but they're not hyphae. And they're dimorphic. Some fungi can take on both the yeast form and the mold form. And usually the yeast form is the more pathogenic, and it forms and lives in our body temperature, so like 37 degrees Celsius. But the mold form usually forms and is pleasant, and it's not as damaging to us. And it's actually a decomposer at temperatures that are cooler, like 25 degrees Celsius, so like room temperature. So the yeast, you can see, has a ring, kind of an oval shape to it. And why I say that they're kind of irregular in shape, because when they bud off, which is their way of asexual reproduction, each bud will form kind of a bud scar, and you can see those on the surface, and they kind of look irregular. So they're not perfect oval, and they're not perfect rod shape. They're just unusual and irregular in their shape, but they're a single cell. And so pseudohyphae form when several buds stay attached and they form a chain like that. So how do fungi get their food? They are heterotrophic. They do not do photosynthesis, so they get their nutrients from a variety of other substrates. And the other feeder is what heterotrophic means. So hetero means other, and troph means feeding. And so if they're an autotroph, they're self-feeding. They get their food from, photosynthesis. And so heterotrophs have to acquire their nutrients. So most of the fungi are saprobes, which mean they get their food from dead plants and animals. So they're detritivores is what we would call them, or saprobes. So they live off of dead things, and they're a good decomposer, and we need those decomposers out there. The problem is when they're trying to decompose live bodies, they're trying to decompose us, but we're not dead yet, and that's when they cause damage and disease. And so parasites do harm to living organisms like plants and animals and other types. They can form on all kinds of living organisms. So they can be parasites when they live off of living organisms instead of dead things. So you can see anything from raspberries. This picture is so weird. I could, until I tell you that that's raspberries, you would be, what is that we're looking at? But these are fungi. And if you've ever purchased raspberries at the grocery store, you look at them before you buy them, and they're perfect and everything. And by the time you get them home and you start washing them, they already have mold. They already have fungi on them. So they definitely can feed off of things like strawberries and raspberries and other kinds of fruits and vegetables. They can also live off of you, and this would be like parasitic athlete's foot, for example. So we are nutritional sources for the fungi when we have athlete's foot. So microscopic fungi grow in loose associations or colonies. The mycelium are where you have a mass of hyphae or a lawn of intertwining hyphae that make up the body or colony of a mold. Septa are, some hyphae have these cross walls that divide the hyphae into segments, and spores form, they are the reproductive bodies. They come off of the different mycelia, and sometimes they can be sexual spores, and sometimes they can, so they go through like a meiosis type thing to form spores, and then they're 1N spores, and 1N combines with 1N, meaning half the number of chromosomes, and they form a 2N, which has the full complement of chromosomes for the cell. So that's one, they can be asexual or they can be sexually reproducing fungal spores. So here you can see spores being produced by these reproductive hyphae, the sporangium, which is kind of like a thin membrane around them, and then the thin membrane breaks and releases the spores, which go and land on the food, and then they form hyphae. They germinate, they form a germ tube, and then the hyphae becomes longer, and eventually they come off out of the inside and break through the surface. And then that's the vegetative hyphae. So that's a reproductive hyphae, and this is just a vegetative hyphae. And that's what it looks like on bread. So this is an example of a bread mold type thing where they have those sporangium. And the reproductive, again, they have lots of complex, successful reproductive strategies. They can just simply grow outward and exist as hyphae. They can fragment off and generate a whole new colony. They can form spores, so they should not be confused with the endospores that bacterial cells produce. Bacterial cells produce an endospore, and then it kind of breaks open, goes through germination, and it produces a single cell. So these spores are a method of reproduction, and it's their primary method of reproduction is spore formation, and they're released out of the cells. In fact, the spores themselves are a cell, but endospores can germinate and become a cell, but they are formed as a survival mechanism, and we'll get into that more in chapter 4. So asexual spores, like the sporangiospores, they form a thin membrane around, and they are formed by successive cleavages within the sporangium, so they form lots of spores from one cell. And then these conidiospores can just kind of break open and break off and fragment off. So conidiospores are not covered. Sporangiospores are covered, and that would be kind of the difference between those. But these are both asexual type of spores. sexual spores, they go through an meiosis type process and produce half the genetic material, so that when they fuse with another cell which has half the genetic material, they can make a new cell. And so most of the fungi have both a sexual and an asexual type of spore that they can produce. There is one type of fungi that's called the imperfect fungi, and the only thing we know of right now is that they have an asexual type spore. And so they don't have a known sexual spore. But I would say that it's probably just that we don't know what their sexual spore is. We haven't identified their sexual spore as of yet. And so the variations in the sexual spores are how we a lot of times will identify their groups or divisions of fungi. That's kind of how we classify them, is by their sexual spore. But they're important because they give us variations, because, you know, just like asexual reproduction just produces exact copies, but sexual reproduction, you combine two organisms and you get a new organism, a recombinant organism, which is half genes from the mother and half from the father, which, you know, it's not like they have male or female fungi, but they have two different organisms that combine together and make new organism with variations of the parental fungi. So slight added variations in the form allow them to survive better. So sexual reproduction actually gives the organisms an advantage, because they're not going to be wiped out as quite so easily because they have such variations. So fungi identification and cultivation, they can look at them microscopic and macroscopically. They look for their asexual and their sexual spores to help identify the genus and species. They look at the hyphae type, the colony texture, the pigmentation, the physiological characteristics and genetic makeup. But overall, to classify them into their divisions, which is the subgroup of the kingdom fungi, we use the sexual spores. So some representative fungi. Here's a bread mold, and this would be maybe Aspergillus right here. So those are a few. And there's definitely some pathogenic fungi out there. A lot of disease, you know, we think most diseases are either bacterial or viral, but there are quite a few diseases caused by fungi, and they can mimic symptoms that would be caused by a respiratory disease, for example, caused by a virus or a bacteria cell. And so we may treat them with antivirals or antibacterials, like antibiotics, and then they don't get better. And so there's a lot of community-acquired infections caused by environmental pathogens. There's hospital-acquired infections caused by fungal pathogens in the clinical setting. There are opportunistic infections caused by pathogens infecting already weakened individuals. And so there are definitely a lot of them. And it's usually coming into contact with them through accidental contact with soil, dust, or water. But again, like in a hospital setting, they can be passed on from person to person in a clinical setting. And especially opportunistic infections can happen when they're weakened. Like if you have a bladder infection and you take antibiotics, it wipes out everything bacterial but not the fungi, and so you could get a yeast infection. And that would be some different ways that you can, different kinds of fungi, pathogenic fungi. Other types of medical conditions, allergies, and sometimes they produce toxins that can cause neurological conditions. So like many people are allergic to penicillin, which is produced by a fungi. That's an antibiotic. Other people just have mold allergies in general, and they have the seasonal allergies caused by fungi spores. And neurological conditions, there's a bacteria or a fungi called Stachybotrys, which is that black mold that's found in like areas where there's a lot of moisture, in walls, like drywall. It'll be found a lot of times, and it can produce a neurotoxin that can be very permanently damaging to the brain. So those are some of the detrimental things. So pathogenic fungi, allergies, and toxins that they produce. Beneficial things, though, and again, we always have to remember that they are here for a reason. Fungi are necessary for a lot of different reasons. Decomposition is the main one, but they have stable associations with plant roots, and they increase water and nutrient absorption. So there's these things called mycorrhizal, roots that are kind of like embedded with fungi that help increase water and nutrient absorption by plants. So plants really need fungi. Decomposition is really necessary, or else we'd be up to our eyeballs in dead things, and we don't want that. They also produce good things for us, like antibiotics. A lot of antibiotics are either produced by penicillium and cephalosporium, which are fungi. Alcohol is produced by fungi. Organic acids are produced by fungi. And vitamins are produced by fungi. And they are necessary for food flavoring. And, of course, alcohol and wine and the gas that causes bread to rise are all benefits of fungi. So not all fungi are bad, and there are definitely some benefits of fungi. So here's the concept check for the fungi, and here are some of the questions. A blank is a type of fungus that derives nutrients from dead or decaying material, and that would be a saprobe. True or false, fungal spores have a tough outer coating that allows them to survive in extreme conditions. So fungal spores, that is not the case. This is false. What they're talking about here is the spore coating of an endospore in bacteria. Okay, the third one, blank are asexual spores that contain a sac-like head. That would be sporangium or sporangiospores that are going to be in that sac-like structure. The conidiaspores don't have a sac around them. They're naked. They have no, it's like, that's the root word, conidia means naked. So they're naked spores. They don't have a sac around them. True or false, the pathogenic fungi require a host to complete their life cycle. So if they're pathogenic, that means that they are in a parasitic relationship, so they need a host to go through and cause disease. So that would be true. And that's the end of section 4.